Yes, sir.
Shall I start, Shanta?
Yes, sir. Yes,
okay.
Sir.
Okay, so thanks to Shanta Rajlakshmi and the HOD of the chemistry department and to the whole Parul University for inviting me for this talk. So before starting, I have a request actually. Can you, the one who are using mobile phone, can they make it in that landscape mode so that my slides will be like, they can see in a better form? Okay. So my name is Mohammed Ithar and I'm a computational chemist. And I have spent most of the time in modeling chemical molecules and their applications in material sciences and also for their medicinal implications. So now I am working in University of Cagliari, which is situated in Sardinia. This place is quite famous for white sandy beaches, blue zones, greenery, and almost zero pollution. We have longer lifespan. The place is having world's maximum 100+ year old population. So I can see many old faces here. And this is, here the university is. So now concerning today's webinar, I will attempt to give a basic understanding for the students by providing a holistic view of the field of computational science. So with computational science, I mean computational chemistry, biology, or bioinformatics. Okay. Am I audible?
Yes, sir, you are audible.
Okay, there's some confusion. Sorry for that. So in this presentation, we will be covering an overview of the computational science and molecular modeling field. And then I will tell you what all can be done with computational chemistry, and then a few case studies that I have worked on, and then we'll be taking questions after that. Yeah, so I guess we all are very much convinced with me that we are living in techno age. So most things and immense information is just one click from us, and everything is moving towards simplicity so fast. So you can see many new technologies are evolving each day. So either we have realized or not, but the truth is we are so obsessed and surrounded with the technology. You can think of few examples like I have quoted here, like Alexa and Google devices. You just need to give them instruction and then they will play a nice song for you. Like Bitcoin, blockchain technology, 3D printing. You can even design your three-dimensional office or home just by printers in no time. We have quantum computing, we have facial recognition devices. All things are very much evolving with so much technology. So we are very much obsessed with that. So now similarly things have also been transforming in academia, and now chemists and biologists are also adopting the evolving technologies of these new approaches to smartly transform and their bench work. I think you are very much somewhere heard about these terms like algorithms, data science, artificial intelligence, virtual screening, coding, programming. So these are, very popular terms nowadays. And even scientific journals are also now flooding with these newer methods and technologies. I have taken few snapshots of these like recently published journal. You can see how this technology is evolving. Artificial intelligence everywhere in drug design, in polymer design, in material sciences, in designing solar cells. It's everywhere. So you can recognize the potential of this field. Even nowadays, like a lot of funds has been spent in this field. Machine learning, polymer memory, everything. Everywhere, it's everywhere. Now, all these technologies and terms looks highly sophisticated and complex, but it's fairly simple. Let me take you to your school days when you have started studying science. We have been taught that science comprises these four parts, which starts from observing some processes, like famous Isaac Newton's falling apple story, and he discovered the law of gravitation after that. So once you have observation, you experiment and check its redundancy, and then we later conceptualize and make a theory. So once you have a consolidated theory, we can then predict the formula. We can then predict the experiment just by a formula, like law of gravitation, like speed is equal to distance upon time. We have one parameter, and then you can compute the other one. So the idea is experiment can be better understood when you have a theoretical knowledge associated with it, right? So once you have established a theory, you can directly go for predictions, and your prediction will depend upon the accuracy of that theory. So whatever science we are doing, we all are sitting somewhere at one arm of this triangle. So I think there is, I mean, sorry, there's a lot of confusion. Can you, I mean, mute, I mean, hello? Hello?
सुरभि मैडम ने क्यों शेयर जो सुरभि मैडम को पूछो ना उन्होंने कैसे किया हाँ
Okay, let me start.
हाँ हो रहा है हाँ सर मेरा हो रहा है
Okay, let me start. So
okay,
Other than sir, can everyone mute themselves? Kindly mute yourselves.
okay, I think it's better. Thanks, Shanta. Okay, so the idea is that experiments can be better understood when you have a theoretical knowledge associated with it, right? So once you have a theory, you can directly go for predictions. So whatever science we are doing, either we are in biology or life science or industrial science or a computational science, we are doing science either sitting at one of the triangle. So we are doing some experiment, some computation, but we need support from the other side.
कौशल भाग्यश्री और ध्रुवी उनके एग्जाम फॉर्म का इश्यू है ना इसलिए वही गए 5 मिनट
Everyone, please mute your mobile phones. Okay. Those who are in lab, students and faculties, please mute your mobile phones. All the students and faculties are requested to mute their mobile phones.
It's okay. We can go ahead.
Okay.
Yeah. So this is how the field is. So we are, I mean, either we are working this side or that side, we are somehow complementing each other. So that's how the science evolves. Yeah. So now we have been refined mathematical, we have very much refined mathematical theories, we have good fundamentals. And every day we have data coming up from different experimental observations. So it will not be an unrealistic thing if I'll say there will be a time when we will ask most of our scientific questions from our computers, and then a computational chemist or a biologist or a robotic computer will simulate and give answer to you. Like, hey computer, suggest me a material which can withstand high temperature, and then computer will simulate. And then, I mean, there may be another question, there may be biological questions, there will be material science questions. I mean, there will be a time then we will be having most of the things associated with the computational science. Like you can ask for the transition state of my reaction. Like say your reaction is not working, you can search for a catalyst. Like you can predict like at what temperature, at what pH my reaction will go, and what will be the yield, what will be the product. So those kind of things are very much evolving day by day. And this is how this field is evolving. And this field is growing immensely, and most of the application-oriented developments had come up very recently. I mean, during this COVID time and during this last 10 years, this field has evolved quite a lot. And this is how the field is. So just realizing the utility of the chemistry, till now, even 2 Nobel Prizes in chemistry have been given in computational science. One Nobel Prize was given in 1998 to John Pople and Kohn for the development of density functional theory and the formulation of this Gaussian. And even Gaussian software is also the follow-up of that. And the second Nobel Prize very recently has been awarded to these 3 young gentlemen. And then it was for the development of powerful codes you can simulate reactions. They have embedded quantum mechanics and molecular mechanics, and then now with that formulas you can calculate chemical reactions, how they transform. So with computational science here, I mean all the like bioinformatics, computational chemistry, or computational biology. So I'm merging all these things all together in order to explain, give you a holistic overview to all the students. So computational chemistry or biology is a set of techniques for investigating chemical or biological problems on computer rather than going into the chemical or real life experiments. So this has so you have a results of theoretical chemistry. You can incorporate those results into computer programs, and then you can compute the properties of your interest. So this is how the field is. So the reason why computational models are so famous among scientists is because of its simplicity. They are very much easy to visualize and easy to rationalize. And with a strong backup of the theoretical knowledge, such computational models can play a crucial role in understanding the chemical process or a biological or a biochemical pathways per se. You can think about like this molecule, how molecule is, how the electrostatic potential of my molecule, which side will be negative potential, which side will be positive potential, and then you can think about the reaction mechanism accordingly. Like DNA model, how the unsaturation is, how carbon is bonded with another carbon, and how the helix is. Like you can think about like how two molecules of oxygen reacts with methane and form carbon dioxide. So how in which side and how the reaction facile. Like brain signaling, how the gene transcription is, how signal network is. So even molecular reactivity. So it's very easy to rationalize and easy to understand and make the things more simpler. So in other words, like more the observational data, clearer will be the understanding and knowledge, and thereby better will be your model, which can make your precise predictions. So having observational data may not be the case every time. So even if you don't have background data, we can still make the predictions using state-of-the-art computational chemistry methods and codes. So even most of the computational chemistry methods are driven by molecular mechanics or quantum mechanics. I think you must have been taught all these things in physical chemistry course. So this is just the application of that physical chemistry equations in order to simulate the chemical or biological processes. So quantum mechanics and molecular mechanics. So molecular mechanics is based on the Hooke's law, and they just treat atoms as if they are connected each other by spring with a force constant. So owing to its simplicity, they are pretty fast, and you can implement these methods for bigger systems like proteins, your multimolecular assembly, your biocomposites. So big systems can be easily be simulated. Now, whereas quantum chemical methods like Schrodinger equation, it considers the electronic structure, and for this reason, they are more accurate, but they can only be applied for smaller systems. So these are about the methods which are already there with your software program. Yeah, so there are many other mathematical models embedded in the program. We don't need to know everything out of it. We just need to understand like how it is, and then we can simply go with the click of the software. So basically, these are the general investigation in computational chemistry that we do day by day. Like you can optimize the geometry of your molecule. You can see like how my molecule is like according to a whisper theory or covalent bonding or the like bonding models. You can easily see how the molecule will look like if this has this kind of bonding and this has this kind of bonding. What will be my bond angle, and what will be what will be like the possible reaction that it can go. And also you can also visualize, you can also understand the transition state of the molecule. It's pretty simple, it's very easy. Just by loading the molecule, you can run a code and then it will give you the transition state. So and then you can also identify the chemical reactivity of the molecule. You can easily see the electrostatic potentials, where the nucleophilic site is, where the electrophilic site is, and then accordingly where the reaction will proceed. And not only that, you can also do thermodynamic calculation, like you can calculate free energy, entropy, and other molecular, thermodynamic parameters easily. Not only that, you can also do protein calculations, like in drug design and discovery that we do, docking or molecular dynamic simulations that are fairly simple nowadays. And also spectral calculation also you can do, like IR, UV, NMR, and accordingly you can predict the photochemical properties of your molecule, of your system. Sometime in experiments also we do compare with the computational data in order to verify our results. So this is how the routine investigation, and then I will take you like one point for each point. So like molecular orbital, you can easily visualize like how will be my HOMO, how will be my LUMO. Like in carbon dioxide, your HOMO will be situated on this side, and then when you have a LUMO, you will be having a node, and then accordingly you can easily understand like how what will be the reactivity pattern of my molecule, where and how the electron resides. And you can also do a scan of your molecular geometry. Like if you extend any bond or dihedral, what will be the minimum of that bond. So let's say if you extend this bond, if you extend this dihedral, what will be the minimum energy conformation? So based on the potential energy scale, you can identify the conformational geometry, conformational behavior of the molecules. And then transition state. So so during the course of the reaction, the structure of initial molecule changes to give the final structure, which is the product. So now this transformation path leads through the hill, where the highly unstable transition state is formed. So this transition state is not a stable molecule and cannot be isolated from a reaction mixture. However, it can be located using the theoretical chemistry, and then you can give an insight into the reaction mechanism. So you can calculate the transition state. Not only that, reaction energy profiling as well as activation energy barrier is quite simple to calculate with modern programs. Even there are programs where you just need to give an input with the specified reaction conditions, and it will generate all the possibilities which can help a chemist in decoding their reaction. So thermodynamics of a process or a chemical reaction, we can also simulate. So in the form of enthalpy or entropy change, and using these partition function concepts, which are already embedded in the software program, you can calculate these parameters. Like I'm giving here an example of like how ethyl radical abstract a hydrogen atom from the molecular hydrogen, and you can calculate the enthalpy and the free energy change. You can see here it's in positive, so this energy is not in forward direction, this reaction is not feasible. So these kind of like feasibility experiments also you can do for your reactions. So now I will give you a glimpse of the field from the computational biologist or a computational medicinal chemist perspective. So so you can think like if you just switch carbon, nitrogen, oxygen, and sulfur, you can have 10 raised to the power 63 combination of the molecules. So this much is the bigger chemical space just by having 30 combination. So this is our expectation, like we should have this much chemical space. And you can think about this chemical space just by imagining that whatever stars we have, like in the universe, that is only 10 raised to the power 24. So we can have a combination of, 2 times as the number of stars that we have in the universe. And now what do we have in our inventory list? We have these many combination, 10 raised to the power 20 combinations. So like we are only have the access of this much chemical space, but we can have this much chemical space. So now with experimental chemistry, you can't synthesize all the molecules and you can't have this much bigger chemical space. But this limitation is not there with computational chemistry. You can always go back to your this much chemical space, and then you can screen a lot of your molecules for a small protein target, and then you can have a few finalized compounds, and those few finalized compounds you can take into the experimental chemistry, and then you can screen those compounds. So this is the beauty of computational chemistry. You can you can screen all possible compounds and speed up the cycle and make the things more efficient in drug discovery. So this COVID time has turned out to be a major outbreak in this field. And actually this time, that shift has been shifted. The spotlight is there for the computational drug design and the computational chemistry. So I mean, a lot of efforts have been given on the drug repurposing, like whatever drugs we already have, can we check them for any other applications. I mean, many compounds are in clinical trials for these appraisals, and also like for especially for COVID development, drug development, like most of the efforts have been initiated from the computational perspective. And there are a lot of gene networking mapping analysis also are undergoing, and a lot of development has been come in recently in this field. Actually, computational chemistry, especially computational biology, the deep learning and machine learning methods are majorly like implemented during this time. So we can assess the targetability of a particular target. Either we should go for this one or should not go. Those kind of initial experiments we can easily do with computational methods that we have. And one very recent example of the drug that came out from the computational design is this baricitinib. So using the artificial intelligence and the computational tools, this drug has been designed, and it is being implemented for the COVID drug, anti-COVID drug. Yeah. So realizing these potential, even US administration has also shown interest in incorporating more data to improvise the computational method by appropriate policies. So a lot of development has been undergoing in this. So now if you are someone who is working in drug discovery field, you must be using this pipeline in order to design a chemical compounds against any disease. So either you can start from a small molecule or the databases that are there. So we have like a lot of databases that have, that have a lot of compounds. You can start looking for the drug from that database. You can prepare the ligand. You can go for ligand-based approaches. You can go for structure-based approaches. Ligand-based approaches like pharmacophore, QSAR. You just need an information of the ligand only. You can start the pipeline, or else you can go with a structure-based approach, and then you can go for the lead optimization, and then you can go for the further optimization of the drug, like assessing the AD-MET profile, PK/PD profile, and like that. And also, if you want to go for a structure-based approach, you can get your protein from the RCSP database, protein structure database. From there, you can get a target structure. You have a ligand, and then you can go for a docking, and then you can assess the affinity, and then you can further test the stability of the complex. And it's not only limited to that. We can also do a fragment-based drug designing. We know some fragment bind to this pocket, and then we can optimize that fragment using a scaffold hopping or de novo design. These are very sophisticated and recent approaches that are coming up nowadays. You can simply assess the pocket of the protein, and then you can screen based on the fragments, and then you can build full molecule from the fragments. And this is being, nicely worked out in many drug discovery processes, which I also have been involved with. Okay, so now, as I told you, there is immense data available in the public domain. Even while sitting at home, we can start a small drug design project. So there are freely available databases like Sync, PDB, ChemSpider, Drug Bank. There is a huge list. I have just given here a few, and then you can take the compounds from here, and then you can start docking with these freewares, with these docking programs. And then also you need to know the active site where your compound will bind. You can take the compound, you can start the docking using a particular site if you are aware with, or else you can also predict that site. And then if you have fewer compounds, you can go for the AD-MET toxicity, and then later on you can take them into experimental screening. So basically, in computational designing, the one of the major approach is molecular docking. So molecular docking is actually a molecular recognition process. So it's a process of identifying how molecule bind and do crosstalk. So this kind of crosstalk or this kind of molecular interaction is very important in events like transcription, translation, or signal transduction or catalysis. So you can simply search in the receptor how your ligand bind, and if that ligand bind, what is the score of that, how is the affinity. If the affinity is good enough, you can list it out, and then like that you will have a list of compound, and then further post optimization or post validation, you can go, you can test, and then you can go for further optimization. So ideally, from docking, we can get a score, and also we can get a pose, like in which what kind of interaction it will make. If that interaction is in reference with your reference molecule, then it's good enough, and then you can select it. Otherwise, you can reject it. So, so in the drug discovery process, it's quite useful. You can screen a lot of compounds, like millions of compounds for a protein target, and then it will save your time, resources, and cost. So essentially, molecular docking is based on two models, which is lock and key. Like most of us have studied in our 12th class level, this lock and key model is a kind of enzyme substrate model. Substrate will bind to the enzyme in a certain fashion, and then it will block the activity of your enzyme. If it is a competitive inhibitor, if it's non-competitive inhibitor, it will bind somewhere else, and then it will affect the activity. So sometimes it's a kind of very rigid binding, and sometimes it's a kind of induced binding. When the substrate will bind to the enzyme, there will be a conformational change, and then that conformational change will have a biological effect. So we have a model based on this kind of binding, based on this kind of binding in docking. So accordingly, based on the protein target, based on the biology of the protein target, we can select a docking model and then go ahead for the docking. So molecular dynamics. So molecular dynamics is one of the most popular method whereby you can actually see the dynamics of chemical or a biological system with the evolution of time. For example, you have a heterogeneous system of lipid. You can actually see how the lipid bilayer will form. You can see the molecules are coming in contact, and then hydrophobically they are interacting, and then they are forming a bilayer membrane. And this looks very sophisticated, but it's very simple. You just need to make a system and then run for a molecular dynamics, and then you can see these kind of interactions easily. Like protein ligand complex. So you want to see the stability of your ligand with the protein. You can run a dynamics and you can see whether your ligand is really stable in that pocket or not. If that it is stable, it is binding, then what will happen to the protein, and how this biological implication, I mean, how could be the biological implication, or like how this protein can interact with the other stuff to make the biological effect. And then this is a SARS-CoV-2 molecular dynamic simulation. So they have seen like how the spike protein interacts each other, and how the addition process takes place. So these methods looks, I mean, quite sophisticated, but they are fairly simple. They are just the application of Newton's second law of motion, which is F is equal to ma. So basically, based on the atomic mass and velocity, you can predict the trajectory of the molecule or of your system. So that's it's all about the molecular dynamics. So it's a simple application of the Newton's law. Yeah. So up to here, I have given a very broad overview of the field. Now, I will tell you about my field and what and where we are contributing in this field. So basically, I did my PhD in supramolecular chemistry. I started my career of computational science from here. And then I moved to biology in NCBS. I did protein interaction, protein ligand designing, peptide design, small molecule design, SAR, crystal structure analysis, binding energy, all those kind of stuff I have done. So I did little biology here. And then again, I moved to physical chemistry in IIT Madras. I did all the quantum mechanical modeling, and now I'm into biophysics. So you can say I have little more idea of this field. now I'm also using these coding things in order to automatize the process. So here for today's presentation, I will present 3 case studies from a mix of kind of computational biology and computational chemistry. Case 1 is the peptide design, which I did in NCBS working with National University of Singapore collaboration. So herein we were trying to target this SAL4-NURD protein interaction. So essentially SAL4 is an embryonic stem cell and is essential in embryo development for, in morphogenesis. So studies have found that its expression is absent in most tissues, but gets activated in cancer tissues. So one study has reported that this expression is the cause of 55% hepatocellular carcinoma, which does not have any effective treatment till now. So overall, SAL4 has 2 main functions. One is transcription activation of embryonic cell. And second, which is more important for this study, is the repressor of the tumor suppressor gene by recruiting this complex. So for the second function, SAL4 interacts with this, so SAL4 is a protein which interacts with the another protein. And then this inhibition, so this is inhibiting the activation of this, this gene. Therefore, once we disrupt this protein-protein interaction, we can have a strategy for targeting the cancer. So this picture will be more clear, will make you more clear. So this is a SAL4, this is interacting with this complex. So once you inhibit this complex, there will not be any activation of this gene, this tumor suppressor gene, and then you will not be having cancer once this complex is disrupted. So I'll tell you what was the proposal. The proposal was to design a shorter peptide so that we can disrupt this complex. We were already having a bigger peptide, which was around 12 mer. 12 mer means 12 sequence peptide was there, which was binding to this complex. But we want a smaller peptide so that we can design, we can disrupt the interaction using a smaller peptide. So this the bigger peptide was having 23 nanomolar affinity, but we want a smaller peptide that can also have a similar kind of affinity. So this is the binding of this bigger peptide. So this binding is essentially a charged interaction. So basically this has glutamic acid, and then arginine interaction. So this is ionic interaction. So we want to trim this peptide to make it shorter, and then also having a similar kind of affinity. So this was an idea. So, and then we did computational experiment. First, we checked which residue is having how much contribution with the protein. So this is how, so basically this arginine and lysine was making most of the contacts, and most of the binding is coming up from this. So we cannot change these 3 residues. And then we did a molecular dynamic simulation to see which residue is more stable. And if the residue is not very stable, we can change or we can modify that residue with another residue so that we can have a shorter peptide. So in order to design a shorter peptide, we worked on this plan of action. So for checking mutation, so that we can change any residue, and then we can have a shorter peptide of equal affinity. So for this mutation studies we did, and then for mutation we have used 3 approaches. First is the GPSA calculation, which is a kind of free energy calculation that uses solvation contribution. And then second is the FoldX. Like FoldX is another program which has its own scoring function. You can check if you change this mutation, if you change this residue with another residue, if you change this residue with another residue, what will be the contribution? So before making all these predictions, we used to do some control experiments, comparing with the experimental results and the available literature. So we did that. So 2 types of control experiments were done. One is GPSA calculation, which is a replica of experimental peptide truncation. So we made a correlation between the binding energy and IC50 values. And then we went ahead and checked the contribution from the anchoring path, which is RRKQ, this one, the anchoring path. And this anchoring path itself was having most of the energy, most of the binding energy. However, we also compared the results of experimental peptide substitutions. And then you can see first column shows the wild type position of this SAL4 peptide, and the second one are the substitution that checked with the experiments. So you can see the correlation, which is around 90% with the experiment. So if we change this, what will happen and what we have seen in the experiment. So there was a like kind of correlation. So we are confirmed that our computational method is working nicely. So then we went ahead and then here I'm showing you the approach, one of the approach that we used, although we have used many approach. So it's kind of like when you change this serine to isoleucine, what will be the change? So if there is the change is in positive, that means that the mutation is not feasible. If it is negative in energy, that means that the other peptide is more stable. So this is a kind of like GPSA free energy calculation that we did by changing one residue with the other one, and then we designed few combinations, and these were the 7 designs that we finalized. And then out of these 7 designs, these for peptide design, we experimentally designed the peptides, and then we checked those peptides in the ITC experiments at our in-house experiments. And in order to see the stability of these peptides, we did molecular dynamic simulations to see how the protein, how the amino acids, how these residues are orienting into the protein. Are they really making any stable contacts? Are they really stable for a longer time or not? So we have a phenylalanine. Phenylalanine is having a side chain. So the side chain was not very stable. And then we optimized that side chain. And accordingly, we have optimized the other orientation of the protein. You can see here, this peptide is quite stable with the protein. And then it was making all the essential contact that was in reference. So this is one combination. This is the another combination. We modified this position, the first position with the tyrosine. And then this tyrosine was also stable and was having an affinity of 120, which is equivalent to the wild type. That means this is a shorter peptide, only 5 sequence. And then we have one additional residue this side, and then still we can have a similar kind of binding with a shorter peptide. And then we also have designed one more peptide with a methionine substitution. And this is how it was interacting with the protein. It was going into a shallow pocket. So these were the few peptide design that we finalized. And then we checked with the ITC experiments. And then in ITC experiment, which I have not shown here, in the ITC experiment, this peptide also worked nicely. And now we are using those peptides in our in-house experiments in NCPS. So this is one of the successful case study that I have been worked with. And then there is another protein-protein interaction study. So these are the 2 proteins. One is NAF protein. NAF protein actually is HIV protein. So it essentially it do kind of like evasion by relocating MHC and CD80 co-stimulatory molecule. When you have any infection, NAF protein will call these co-stimulatory molecule, and then it will try to check that infection process. So the idea was we have a NAF protein that interacts with the another protein, which is CD80. We have all the biological details and evidences, but we don't know any structure details of it. So what we did in this case study is that we built model of this protein, we built model of the another protein, and then we have our experimental data. And then according to that experimental data, we verified our binding modes. So you can see we don't have any even similar homology structure of the protein that we have in our lab. So what we did, so this is all our unstructured, we don't have any nearby protein. So what we did, so we don't have any information of this part, we don't have information of this part. So we have anchor domain, we have core domain, which were unstructured. So what we did, we did homology modeling. So in homology modeling, I mean, there can be 3 kind of dimerization interfaces this protein can have according to the literature details. So we build kind of consensus model for that protein. And then this was a finalized model. And then also for the another peptide, which this protein interacts with, for that one also, we don't have any model. And then we model that using homology modeling. And then this was the experimental NMR spectra NMR chemical shift perturbation data that we have in-house. We know this protein binds, we know these residue are interacting with, and then we map these residues onto our protein. And you can see this is a site, these are the sites, this are the residue which were falling into. And then only to these specific site, we did docking experiments, we did dynamic experiment, and this was the final binding mode that we came up. And this was the validated binding mode because we have all the experimental proofs to show, to establish that this is actually which is happening in the biological scenario. This is the third case study. So this is about the computational, mainly like towards the chemistry side. So here actually, so what we are trying to determine, or what we have shown here, that we have a fullerene, which is N-fulleropyrrolidine. So we have a pyrrolidine substitution on the fullerene, and this NFP, we call it NFP, can bind with pyruvate, and then there will be a supramolecular aggregate, and then that supramolecular aggregate will be fluorescent, and you can see, and you can see them in experimental conditions also. So what was the idea? To see the mechanism, how they are binding each other, and what kind of binding lead to this kind of supramolecular aggregation. So this is kind of like, there will be a hydrogen bonding, and pi interaction. So this will be a hydrogen bonding from this side, and this hydrogen bonding will induce the pi stack. So this is a kind of like through bond, through pi interaction. There will be a through bond interaction, there will be a through pi interaction. So this is a newer concept kind of. So this will make a kind of very stable assembly, which was 43.47, which is a very stable complex. And because of that much, bigger stability, this is making kind of like supramolecular like arrangements, and that will lead to fractal kind of assembly. We have simulated them in computational molecular dynamic simulations also. We have simulated them, but before that we did some experiments to see, why these fractals are charged. So we have seen a charge transfer band. So then when we deconvolute this charge transfer band, we found it is made up of two further bands. So charge transfer 1 and charge transfer 2 band. And then we further try to identify like from where this band is coming up. So then we see that the same index in the computation, we are also having a similar kind of shift at this lambda max. And this was like from this hydrogen bond. So the electron density is shifted from this hydrogen bonding counterpart to the fullerene. So this is my donor, this is my acceptor. So it is a kind of through bond interaction. So from hydrogen bonding, so through hydrogen bonding, this is shifting. And this side, so we have an electron density of on pyrene and on this one. So there is a kind of like pi interaction. So this is a kind of like through space and then through pi stack. So two kind of interactions are mainly happening in my complex, that is leading to this kind of assembly. And then we further try to simulate in molecular dynamics simulation, so to see how this aggregation is happening. And then we were able to see similar kind of results here also, and which were quite fascinating to me. And then we try to see like how this complex is orienting and how this complex is making interaction with the another one. So mainly these are making with kind of trimeric complex, basically stabilized with the pi interactions. And then there was a kind of cation pi interactions also. And then there's kind of side bridge interaction. And then they were in contact around like 10 to 15 angstrom, like this complex is in contact with this one, and that was leading to the fractal assembly. So now coming to the introduction, coming to the summary of my presentation. So today I have like tried to give you an overview of the field, like how the molecular modeling and computational chemistry and what can be the potentials coming up. And then I have shown some case studies from my own work, which is peptide design. We have designed a peptide for a particular protein, and that peptide was working. And then we have used that peptide in order to screen the small molecule. We are kind of like disrupting this interaction, and we are designing a small molecules in order to treat the cancer that is underway as of now. And then based on the NMR QC experiment, we have come up with a with a peptide binding mode, and now we know which amino acid and how can we disrupt this interaction in order to take it to the larger scale. And also now this is a third case study from the supramolecular aggregation, and we also have an experimental proof to show that this is happening, and then we have rationalized the through bond through space interaction, and recently we have communicated, and this has been published in ACS. So now I hope I would be able to give you some understanding of the field, and now I think the students can make an informed decision for their dissertation or their further career options. So this is my scientific talk as I conclude up to here, and then these are the people involved with me, and I have involved these many people. These are the nice people with whom I have learned a lot. And then thank you, and thanks Shanta and Parul University in general for giving me this opportunity. Thanks. I can have question. You can ask questions if you have. Thank you.
Thank you, sir, for giving us such a valuable time and such a nice presentation. So, as you have rightly said that it's easy to put an experiment if you have some theoretical knowledge about that. So, the floor is open. Any questions from the audience?
Yeah.
Anyone would like to have any questions or would like to ask something? You can do.
Hello, Dr. Mohammed.
Yeah. Yes, sir.
Yeah. Actually, wonderful work. And actually, I have one little questions regarding what we are studying. Actually, we are studying the morphological studies of surface science.
Okay.
And we have synthesized a few novel surfactants, and that we have achieved certain morphology like micelle, ellipsoidal micelle, vesicles, bilayers, helical structures. These are the structures we have already prepared.
Okay,
and,
nice.
And check their morphologies and applications as well. Now we are diverting this our core subject to the application point of view, and in organic chemistry.
Okay,
so is it possible that can we check through computational chemistry whether it is going to be possible our morphology to the organic chemistry?
Yeah, I think to some extent you can. So I mean, it's a kind of like you need to understand like how is the molecular arrangement, like when you have a kind of like molecular model, like how is the setup, how I mean you exactly know the stoichiometry kind of things, like what is the composition of your system. If you have that idea, and then you can take it to the experiment, you can take it to the computational level, and you can see like what can be the possibilities. So kind of like you need to have a like qualitative understanding of the composition of your system. If you have that, then you can try the computational modeling of that. I have worked with one of such project. We were working with the lipids that were making kind of a special morphology.
Similar kind of.
We were having DPPC and cholesterol mixture, and then it was making a kind of like a surface morphology. And then we were kind of see like how this, I mean, we were kind of interested in how the molecular arrangement and what is a kind of like bilayer thickness and this kind of things. Yeah, I have worked with that. And then we were able to show like
actually we are right now synthesizing a new kind of surfactant that is a cholesterol base, and we would like to check whether it is going to form a bilayer or not.
Okay, those kind of thing you can, I guess, if you have
this is possible, I think.
Yeah, of course this is possible. I was having I was not having full understanding of a system. Now I worked with this kind of system, DPPC cholesterol system I have worked with, and then we show the arrangement, how the arrangement is, how they are forming a bilayer, what is the thickness, what is the area, and then the same kind of things. Also there was in Langmuir kind of experiment we have complemented with.
The surface chemistry.
Yeah. I remember that long back. I did that. Yeah, those kind of things you can do. Yeah, there are very well-established computational models for that. We have core screening molecular dynamics model, which are very much implicated for these kind of experiments only, like lipid chemistry. You can check those articles, like core screening models. Yeah, they are very much. Yeah, even now I'm also working in computational, like protein science. Thank you.
Okay, Sure. Thank you. Thank you so much.
Yeah, welcome.
Thank you, sir. Anyone from the audience side?
Even they can ask questions by typing if they are hesitating.
And actually, we professors, we have asked whatever we wanted to know, and PG students are little, young. So
yeah, I can understand. I can understand.
but,
Even
it is
I have
valuable
been.
for their dissertation work. They have just entered in that field, so we just wanted to have them that kind of exposure. So that
I know. Yeah, I was trying to, focus on that part rather than going into my work.
no, sir, it was good. It was a general thing, so it's better for them.
Yeah, thank you.
Okay, thank you, sir.
If they need any help,
Yes.
please continue, Shanta.
Sir, are you planning to visit India
Not very soon.
in December or January?
Maybe
Okay.
December, January. Yeah, maybe I may come in January, December or January. I will be happy to visit your labs.
Surely, sir. So,
Yeah.
we would like to have some talk kind of a session from your side.
Yeah.
Would it be possible? Then we'll definitely communicate for that.
Yeah. That will be good. Like live interaction will be like
Yes,
much more
definitely.
conceivable.
Yes,
Yeah.
sir. So I'm winding up the session here only.
Yeah.
I think. Thank you once again, sir.
Yeah. Welcome.
Okay. Thank you, sir.
Thank you.
Thank you. Thank you all. Have a nice day. Yes. मैंने तो रिकॉर्डिंग भी की है कंप्यूटर करके। मैंने स्क्रीनशॉट ले लिया और मैंने रिकॉर्डिंग कर दी थी। एक स्क्रीनशॉट अभी ले लो ना, अभी तो निकल गया ना आंसर, अभी कोई मतलब तो वीडियो जब रिकॉर्डिंग ली तो आ गया होगा ना कोई स्टोरेज में। ठीक है, मैंने ले लिया है मैंने। अच्छा था इसका काम।